Hi everyone, today we're studying about electron transport chain, which is oxidative phosphorylation. So electron transport chain is basically a series of complexes which are located on the inner mitochondrial membrane. Now, if you look at the mitochondria, so this is basically a mitochondria. It will have matrix in the middle, and these are its membrane. So you have an outer membrane, and then you have the inner membrane. The inner membrane forms more of a crystal-like structures, right? So between the outer membrane and inner membrane is the intermembrane space. So I hope you're understanding. This layer right here is mitochondrial matrix. Then this membrane is the inner mitochondrial membrane. So it's very important. So I'll just label this right here. This is inner mitochondrial membrane. Here is your intermembranous space, right, which is between the two membranes. Now, electron transport chain is basically a series of complexes which are located on the inner mitochondrial membrane. So these are the series of complexes. You have complex 1, you also have complex 2, then you have complex 3, you have complex 4, and complex 5 is basically your ATP synthase, right? Now, now complex 2 basically does not pump protons, so that's the reason they've not shown it here, but there is complex 2. So let me just draw it on my own. This is complex 2. Now, the order is the first complex is given the name of NADH dehydrogenase. It's given the name of NADH dehydrogenase. And also, what is the coenzyme that it works with? It works with NADH. NADH comes here and it donates an electron. So NADH is an electron donor. Electron got donated. That electron is going to be transported further. So this is your first complex, which has the name of NADH dehydrogenase. Now, then right here, you will have one thing called coenzyme Q or ubiquinone. Now, this is a mobile electron carrier. So what happens is that the electron that came to complex 1, it's going to be picked up by coenzyme Q because it's a mobile electron carrier. And also, then the complex 2, it is given the name of succinate dehydrogenase. So what's the name? Succinate dehydrogenase. Now what does succinate dehydrogenase mean? Now this basically deals with FADH2. So FADH2 comes here and it gets converted to FAD positive, which is oxidized. So basically the reduced FADH, it gets oxidized. Now here also obviously electrons are donated because it's an electron donor. That electron is again taken up by the coenzyme Q, which is the mobile electron carrier. So coenzyme Q is ubiquinone. Now moving forward, that electron is sent to the complex 3. Complex 3 is given the name of, what do you think the complex 3 is called? It's called cytochrome BC. It's written right here. So cytochrome BC is the name for this. Cytochrome BC. Then moving forward, you will have another very important mobile carrier, which is cytochrome C. Right here, it's a mobile carrier. The electrons from here, they're going to go to cytochrome C. So where are those electrons coming from? They're coming from coenzyme Q, right? So they will be sent to cytochrome C. Cytochrome C will carry them to cytochrome 4. Cytochrome 4 is given the name of, since this was cytochrome C, so this is cytochrome C oxidase. So I hope you can recall. The first was NADH dehydrogenase, the second is succinate dehydrogenase, the third is cytochrome BC, the fourth is cytochrome C oxidase, and the fifth is ATP synthase. So now complex 4, which is cytochrome oxidase, it receives the oxygen and what does it? It receives the electrons and what does it do? It sends those electrons to the final electron acceptor. What is the final electron acceptor? It's the oxygen, half an oxygen. So half an oxygen, it accepts that electron and it gets converted to oxygen. So you find it gets converted to water. So the final product is water. So you got the water, and this electron did its work. What was electron's job? Its job was to ensure that protons get accumulated inside exactly where in intermembranous space. So it pumped out protons from the outer mitochondria, from the mitochondrial matrix. From the mitochondrial matrix, those protons got pumped outside into the intermembranous space. So four protons in the complex 1, four protons in complex 3. There are none in complex 2. That's because it doesn't have sufficient, I mean, because there's no energy that's lost in this process of, an electron transport here. So how are you pumping these protons? That's because when electron is being shifted from one complex to the other, there's a certain amount of energy that's being lost. So the energy that's being lost, it's used by this gradient. It's used to develop this gradient. That gradient is where you're sending protons from mitochondrial matrix into the intermembranous space, right? So mitochondrial matrix has abundance of protons. They're being pumped into the intermembranous space. But for second complex, there no electron energy is lost. That's the reason none of the protons are pumped through here. And then here you have four protons. Here also you have two protons. So four, four, and then two. I hope you remember the number of protons. Now what happens next is that these protons, they are going to easily diffuse out. So how many are they? 4 plus 4, 8, and then you have 2, 10. So 10 of the protons, they're going to go from the intermembranous space back into the mitochondrial matrix, and this will be diffusion, and this will happen with the ATP synthase. Now ATP synthase has two parts. It has w- uh, it has one of this. Also, one more thing, this got the name of cytochrome C oxidase, but it also has another name, which is cytochrome A or cytochrome A3. So A plus A3, it has another name. So this is BC, this is A and A3. Well, so ADP synthase, it has two units to it, or it has two parts. One is FO domain and one is F1 domain. FO domain is the one that lies towards the intermembranous space. And so intermembranous space, you have FO domain, right? And then you have F1 domain, which is extramembranous space, or this is in the mitochondrial matrix. So there is an inner mitochondrial membrane. So FO domain, it spans around the inner mitochondrial membrane, and F1 domain, it's in the mitochondrial matrix, so extramembranous. So I hope you remember FO is in the inner mitochondrial membrane. This is something you need to learn. So because of this gradient, what happens that the ATPs are being generated. How? Because that energy is used to convert ADP into ATP, and that ATP is being pumped into intermembranous space. So this is how ATP is being generated. So again, electron flow is first of all from NADH, as NADH is the donor. So from there it will come to complex 1, which is NADH dehydrogenase. From here we'll go to ubiquinone, which is coenzyme Q. Then complex 3, which has the name of cytochrome BC. Then we'll go to cytochrome C. Then it will go to cytochrome 4, which has the name of cytochrome A and A2, or cytochrome C oxidase. And then it will go to finally the electron acceptor, oxygen. And if it's FADH2, so it will go to the complex 2, then coenzyme Q, then 3, then cytochrome C, then 4, and then oxygen. I hope you can understand the entire zigzag pattern. Now, proton pumping is through the first, third, and fourth complex, not in the second, because there is no energy that can be used to drive this gradient, right? So only in complex 1, 3, and 4. So NADH dehydrogenase, cytochrome BC, and cytochrome A3, or you can call it cytochrome C oxidase. Now, proton pumping creates proton gradient. Proton gradient is created, and protons are pumped here. And because of this, there'll be a gradient here where proton will diffuse right into the mitochondrial matrix, and you will get ATPs. And yes, so if you talk, that's the reason when you talk about 4, 4, and 2. So these are basically 10 protons in total. And that's the reason when you talk about NADH, this produces 2.5 ATP, whereas FADH2 produces 1.5, because it started off here and it missed these two complexes. So its energy was generated from these two regions. So 1.5 ATP. Final electron acceptor is oxygen. That's one thing you need to know. If there's no oxygen, that's the reason we say that oxygen is so important, because if there's no oxygen, electron transport chain will stop and it will result in cell death. This is the only reason why oxygen is very important. And also, there's a high yield question where they ask you about inhibitors. So you need to ensure that you know about the inhibitors. I'll just write them down. For complex 1, you have rotenone as the inhibitor. This is about complex 1. For complex 3, you have antimycin A as inhibitor. For complex 4, you have cyanide, or you can say carbon monoxide as inhibitor. And for an ATP synthase, you will have oligomycin. So I hope that's clear. Oligomycin and cyanide and carbon monoxide, antimycin A, and rotenone. That's it.